Okay. Hello, my name is Kalybe VanWatermeulen, and I am currently an undergraduate at the University of Minnesota Twin Cities, studying neuroscience and biochemistry. And this study was the analysis of differentially expressed genes and the deep cerebellar nuclei of 154Q Spinocerebellar Ataxia 1 type mouse models. Some key collaborators were Katherine Hamel, who was the foundation of all the research by doing RNA sequencing on 154Q in wild-type mice, and then seeing the log fold changes in mRNA expression. And then Dr. Maria Cvitanovic, who was my PI and mentor. Two major sources of funding for this project were, one, the Cvitanovic Lab, and two, the Undergraduate Research Opportunities Program, or UROP. So some quick little preface of what to expect for this poster is, one, the background. I'm going to be going into what SCA1 is, what are the symptoms, how is that disease working, how is SCA1 occurring, going into the deep cerebellar nuclei, and then what they are, what they do, and then explaining the 5 genes that I looked at and then some of the related pathways. And then I'll be diving into methods. How did I obtain these DCN samples, or the deep cerebellar nuclei samples? How did I look at the RNA, make the cDNA, and then how did I do qPCR? Next, I'll be looking at figure 1, which is going to be a volcano plot of the RNA sequencing data that was done by Katherine Hamel. The figure I coded myself and is kind of a way to visualize all the genes in the mouse genome that were differentially expressed between 154Q and wild-type mice, and then diving into the qPCR data of the 5 genes that I ran for this experiment. And then going into CNET pathways on the right, figure 7 and 8, just to kind of explore, like, what are these genes, what pathways are these genes related to, what could they possibly be doing, and then ending on future directions and limitations along with conclusions. So basically, we know these genes, we know what pathways they're connected to. How can we prove this? How can we prove these genes, by them being downregulated or upregulated, is affecting these pathways? Okay, so to begin, just a quick little preface. So the study started with RNA sequencing data between 154Q mouse models and wild-type models. As I mentioned previously, that was with Katherine Hamel, who was an old graduate student in Cvitanovic's lab. I have currently identified over 120 differently expressed genes in the deep cerebellar nuclei, and the study just aimed to look at a few of them and their related pathways. So, Spinocerebellar Ataxia type 1, or SCA1, is a progressive neurodegenerative disorder characterized by glutamine, Q repeats, in the Ataxin 1 or ATXN 1 gene. What does this mean? So basically, think of a gene as like a, for those who aren't studying like biology, genetics, biochemistry, for people unaware of this terminology or like what these words mean, a gene is essentially like a sequence of DNA that encodes for protein. And regarding glutamine, there's going to be this trinucleotide sequence, trinucleotide being 3 nucleotides, so like adenine, guanine, cytosine, or thymine. And in the case of for glutamine, the DNA sequence that encodes for glutamine when it goes through transcription and then eventually through translation is going to be CAG, cytosine, adenine, guanine, or a CAG repeat. So if you ever hear about SCA1 or any sort of spinocerebellar ataxia in the future, and you hear CAG repeat, that just means glutamine. Because you go from DNA to mRNA, and then it's still reading that CAG repeat, but it's like the complement. We'll skip the science of that. And then that CAG repeat is encoding for a glutamine amino acid, and then that glutamine amino acid is being incorporated into the protein or the polypeptide. And essentially, so when it comes to spinocerebellar ataxia type 1, you have this ATXN1 gene that is being transcribed and translated, but it has a bunch of these CAG repeats, and these CAG repeats are incredibly sticky. They're going to aggregate to other proteins, other Ataxin-1 gene products, or just anything that's inside of the cell that it can stick to, essentially. It's creating these aggregates, which can eventually lead to cell death or cytotoxicity. In fact, when you look at human models or human SCA1 models, the Purkinje layer of the cerebellum in the SCA1 patients is completely devoid of all Purkinje cells because they're dead. Like, it's wild, actually. Looking at it from a patient who doesn't have SCA1 to someone who does have SCA1, it's just no Purkinje cells, or there's like one or two Purkinje cells. Yeah. Symptoms, of course, it's look at the name, Spinocerebellar ataxia type 1. There's going to be ataxia. Essentially, ataxia is going to be like lack of coordination. Think of like common motor disorders or diseases that you know of, like Parkinson's, Huntington's disease, and stuff like that. Not the same mechanism, different mechanisms, but they can show similar symptoms. And then nystagmus is like another common symptom, which is just like lack of eye coordination. So the deep cerebral nuclei gene expression is understudied in SCA1 models, and in general, the DCN are understudied in general. Doing research for this project was incredibly hard because the genes have been researched in different tissues, but when it comes to the DCN specifically, there is nothing. So I couldn't reference the activity. I couldn't confirm what was going on in the DCN with these genes. Which was a pain, but it's also exciting because now we have a place to dig into in research. How differentially expressed genes or DEGs are present in the DCN could explain how SCA1 models correlate with abnormal behavior. So some previous research in our lab has actually shown that CAG repeats, the size of them correlates positively. So as the CAG repeat increases, or you have more CAG repeats, and that's more glutamine amino acids in your polypeptide, there is an increase in depressive and like anxiety related behavior. So digging into that with this research, and then with like RNA sequencing and then confirming it with qPCR is just very important. And then specifically the DCN consists of the fastigial, interposed, and dentate nuclei. The fastigial nucleus is responsible generally, as all the nuclei are, it's cerebellum, motor coordination, but more specifically eye movement. The interposed nucleus is more so related to stretch reflexes. Muscle spindles and gamma motor neurons are pretty relevant. Muscle spindles being like the things that are muscles that are detecting stretch, or one of the things detecting stretch, and then gamma motor neurons being the things that modify those muscle spindles. Not that relevant, but some cool facts. And then the final nucleus, the dentate nucleus, more so modulating voluntary movements. So think more so the corticospinal tract, if you are more familiar with like neuroscience. It's not directly involved in the corticospinal tract, like it's not one of the main synapses, but it does modulate the corticospinal tract. Anywho, this project aimed to identify key genes that are differentially expressed for numerous pathways in 154Q SCA1 models. And some of the genes that I looked at were CRHR1, which is the corticotropin releasing hormone receptor 1. And essentially, it's doing my research is actually very important in embryonic development, or just development in general. It is also related to cognition and, unsurprisingly, stress. ACTA1 is going to be the alpha-actin-1 protein encoding sequence, and essentially it's going to create actin filaments. It's not really found in the CNS. In fact, I don't think it is found in the CNS, and I'll explain it later in the presentation. But the main reason I wanted to run ACTA1 first was just because of how surprising it was to be in the DCN, because it's usually in the peripheral nervous system, not the central nervous system. PLEC HG4 is going to stand for plextrin homology and Rho GEF domain containing G4 protein encoding gene. Long name, but the main thing you need to know about PLEC HG4 is just that it is like a GEF domain. GEF standing for guanine exchange factor. If you're unfamiliar with what that means, the guanine exchange factor is essentially like a domain that think of like GPCRs or G protein coupled receptors, like how the alpha subunits are going to need to exchange GDP for GTP to activate, or like monomeric ones like RAS and stuff. It's essentially GDP to GTP for some signal cascades. Next one was OASE1G. Also long name, 2' to 5' oligoadenylate synthetase 1G. Main thing to know about it is that it's involved in antiviral immunity and binds to double-stranded RNA. and is essentially just like virus attacks host, virus inserts RNA, and then OAS1G is like, no, we're not going to do that, and then degrades that mRNA so it's not integrated into the genome and then wreaks havoc. And then finally, or no, not finally, excuse me, CST7, the next one is going to be cystanin 5. Its main function is going to be negatively regulating cysteine protease inhibitors. Cysteine proteases being enzymes that are going to break down proteins with a cysteine residue. It's not cleaving at a cysteine residue, but it is, I think it's a serine threonine protease, meaning that it's going to attack there. Don't quote me on that. But essentially, it's going to attack with a cysteine residue, a cysteine protease well. But in the case of cysteine F, it is regulating a cysteine protease inhibitor. So it's not regulating the cysteine protease itself. It's inhibiting the thing that's inhibiting the cysteine protease. And then finally, IL-20RB, or interleukin-20 receptor beta. Again, its main thing is just going to be it inhibits nerve growth factor and has previous studies regarding neuroinflammation and cytokine activity. Cytokine activity being like immune, stress-related, and stuff like that. Yeah. Okay. So, methods. So, how did I obtain these DCN samples, and how did I analyze these DCN samples? So, to begin, I looked at 154Q mouse models and wild-type models, and I sacrificed the mice, extracted their brains, put them into a brain matrix, specifically a sagittal brain matrix. For those who are unaware of what a brain matrix is, it's kind of like this cube that has like a pocket, if you will, that fits the brain inside of it with these slits where you can place razor blades in. And it's basically a way to get gorgeous slices of the brain so you can do what I did, get punches or like specific regions to look at gene expression. Anyway, the punches, I got like 3 or 4 from each one, depending on how good I was. The tissue was very sensitive. Sometimes the razor blades would stick. I would place them into a small tube. Samples that were stored were placed into, or were flash frozen and placed into a negative 80-degree freezer. And the ones that were used underwent RNA isolation through a trizol method. And then with the mRNA that was taken from the DCN punches, I used a reverse transcriptase and some other reagents to create cDNA, and then amplified that cDNA through PCR, and then used that cDNA along with respective primers of the genes that I mentioned in the background to look or compare the relative mRNA expression between 154Q mouse models and wild-type mice. Okay, so figure 1 is going to be a volcano plot and is a visual of RNA sequencing data comparing 154Q and wild-type mice. And then some quick things of note are that pseudogenes are omitted, the FDR significance is less than 0.05, and the log2FC value shows expression change. So, starting off again with a quick background, I made this plot in RStudio based off the data that was obtained by Catherine Hamill, who I previously mentioned. The pseudogenes that were omitted were genes that encoded for nothing useful. When I was doing my research for the gene table that I created of the 120 plus differentially expressed genes, I came across numerous pseudogenes, and I just decided to omit all of them and focus on genes that we have current research on, ones that are more plausible in the onset of SCA1. And the FDR instead of the p-value, some people may be more like, oh, what's an FDR? I use the FDR significance because it takes into account the genome size and is kind of a way, think of it as a corrected p-value. It's a more accurate p-value based on the size of the genome of the organism that we're looking at. And then as for the log2FC value, so yes, it is showing expression change. What does that exactly mean? So for example, if you look at ACTA1 that's on the right of the volcano plot, you'll see that the log2 fold change is going to be around +5. So this is just basically showing that compared to wild type mice, the ACTA1 gene is going to be expressed more or upregulated by a log2 fold change of 5. So very upregulated. Whereas if you look at the genes like OSTN, MYO15, or as I previously mentioned, CRHR1 or IL-20RB, those genes are going to have a negative log 2 fold change or a log 2 FC value, which indicates down regulation, and then the higher the number means higher magnitude of that expression change. And then as for the genes of interest, or the ones that were most important, those genes were the ones that are marked by red dots, which is basically just showing that, okay, so this was significantly changed based on the FDR or corrected p-value, and then the actual expression change itself, again denoted by the log 2 FC value, was also significant. So it was basically, the red dots are 2 significant changes, the green dots or blue dots are 1 significant change, and then the gray dots, which should be most of the genes, are going to be not significant. Okay, so qPCR data, or figures 2 through 6. So figures 2 through 6 show the mean relative mRNA expression of CST7, OAS1G, CRHR1, IL-20RB, and PLEC-HG4, respectively, or the genes that I previously mentioned. Significance was calculated by running a one-way ANOVA test through RStudio, and genes CST7, OAS1G, and CRHR1, their baseline, or like their control group that they were compared to, was 18S. Whereas for IL-20RB and PLEC-HG4, their baseline, or their control that they were compared to, was RPL. And the main reason for the difference in use of control or baseline was that CST7, OAS1G, and CRHR1 were analyzed with cyber green, whereas IL-20RB, PLEC-HG4 were analyzed with DNA probes. So starting with figure 2. So figure 2 showed that CST7 was significantly upregulated in 154Q mice compared to wild-type mice. Each genotype had 10 mice, and then the p-value was less than 0.001. If you look at the figure 2, you'll see that it's characterized by 3 asterisks, and the difference in the mRNA change was about 3. The wild type was used as a baseline, so it should be a value of 1, and the value of CST7 relative mRNA expression was about 4. Figure 3 showed that shows that OAS1G was significantly upregulated in 154Q mice compared to wild-type mice. So instead of 10, each group or each genotype in figure 3 had 8 mice. And the p-value was less than 0.00 or 0.001, again denoted by the triple asterisk. It was a bit less than CST7, about like 3.5-ish, and then the wild type value again was 1, where it should be. Figure 4 shows that CRHR1 was significantly downregulated in 154Q mice compared to wild type mice. The amount of mice for each genotype was 5, and the p-value was less than 0.01, denoted by the double asterisk. Figure 5 shows that IL-20RB was significantly downregulated in 154Q mice compared to wild-type mice. Each genotype only had 3 mice, and the p-value was less than 0.05, denoted by the single asterisk. And then the final figure, figure 6, shows that PLEC-HG4 was significantly downregulated in 154Q mice compared to wild-type mice. And each genotype had 3 mice, and the p-value was less than 0.05, again denoted by the single asterisk. CNUT pathways. So figures 7 and 8 are basically going to be showing CNET plots of the differentially expressed genes based on the RNA sequencing data that I previously mentioned. Please note that the fold change indicates the magnitude of upregulation or downregulation. So for figure 7, we're looking at the pathways learning, cognition, and associative learning. The bigger the node or size, the more genes that are interlocking with that pathway, and the deeper the red in figure 7, the more downregulated that gene is. And learning and cognition is something that is very interesting in the SCA1 models, because previous studies have shown that SCA1 models, their anxiety or like depressive behavior correlates with an increase of that CAG or glutamine repeat on the Ataxin 1 DNA or gene sequence. Figure 8, on the other hand, is a CNET plot for the differentially expressed genes from, again, based on their RNA sequencing data. However, this is looking at an upregulation or a positive fold change in genes. And these ones are mostly related to response to virus or defense response to virus, which is interesting because SCA1 isn't exactly, well, we don't really know the cause, we don't know all those mechanisms, of course, but the body, based on these results, is appearing to upregulate genes as if it's responding to a virus, which is very interesting, very cool. So we have seen that the DCN has relatively no research, and when you bring that into account with the SCA1 model or SCA1 patients, there is nothing, there's basically no research at all. And it's really hard to kind of gauge like what these genes are doing, how can that be traced to SCA1 pathology onset. We've also seen that some genes are upregulated, especially those related to viral response, as if the body is reacting to a virus attack. And we've also seen that some genes are downregulated, such as like CRHR1 or like genes related to pathways connected to learning and cognition. So what are some future directions to kind of elucidate or like help explain or visualize these proposed, alterations between 154Q models and wild type? One future direction would be the immunohistochemistry of the DCN perivesicular nucleus of the thalamus. So the DCN-PF pathway is from the DCN to the perivesicular nucleus of the thalamus, or PF, and it is a glutaminergic pathway. So the DCN is sending a glutaminergic neuron to the thalamus. And what is very special about this pathway is that it actually modulates fear-related behavior. And one thing that I found while researching this pathway is that CRHR1 is integral to this pathway. And if you remember from the qPCR data, CRHR1 is downregulated. So what does this mean with the DCN-PF pathway? Is it degrading? Is it like increase? Like what is happening there? How is CRHR1 affecting this pathway? And then how is that connecting to the, like, as I mentioned in previous studies, the increased anxiety behavior with increased CAG repeats, so that correlation. A second future direction would be the visualization of transcription factor binding. So based on the qPCR data, the relative mRNA expression is being either decreased or increased. How? How is the relative mRNA expression increasing or decreasing? Is the transcription factor binding? Is it not binding? Is it abnormally binding? Is the translational machinery increasing, decreasing? So the mRNA is building up, nothing's happening there. What is happening inside of these 154Q models that is resulting into these differentially expressed genes or these different levels of relative mRNA expression? And then a final direction, pretty straightforward, Western blotting. So this study identified a couple genes and looked at the transcriptional level of gene expression, but we did not confirm the translational level of gene expression. We didn't look at proteins if they were being made, just because the relative mRNA expression of like OAS1 gene was upregulated compared to wild type mice. That doesn't mean the protein expression has increased. So confirming the actual protein products with Western blotting would be very important. And then as for some limitations of this study, major ones to note were just small sample sizes for some genes. As you may have noticed with the, I believe it was the last two, forgot the names, but it was the last two of the qPCR figures. They only had 3 mice for each genotype. That's the bare minimum for qPCR. I would have liked to have at least had like 4 to 6 mice for each one, just to consolidate the results more and have them become more significant. And then another major limitation was that the DCN samples themselves were just incredibly low in concentration. So it was just kind of hard to run these genes, make sure that all the samples were at consistent levels for the qPCR. Yeah. Conclusions. So, as you may have noticed with the qPCR data and the CNET plots and everything else, ACTA1 was not discussed, and that was because ACTA1 was not identified in the DCN. I ran ACTA1 3 times with DCN samples, and then I ran it once with a higher sample, or a higher concentration sample of the cerebellum in general, like the entire cerebellum. and referencing that, the DCN sample from last slide is like 15 to 30 nanograms per microliters of RNA, whereas the ACTA1 or the cerebellum as a whole was around like 500 or more nanograms per microliters, and there was still no ACTA1 expression, which is also weird considering the fact that ACTA1 it had a log twofold change of 5, meaning that it was significant. It was very upregulated. So I would have expected it to be have been seen, but it's also not surprising because previous studies have confirmed that like ACTA1 is a peripheral nervous system related gene, specifically muscle, again actin filaments, whereas it's not found in the central nervous system. It could be there, but again, because we have such low RNA concentration with the DCN, we may not be picking up on it. So possibly to also explore in the future, but that was the main conclusion with ACTA1. OAS1G and CST7 were significantly upregulated in the DCN compared to wild-type mice and 154Q mouse models. And CRHR1, PLEC-HG4, and IL-20RB were significantly downregulated in 154Q mouse models compared to wild-type mice. Okay, so why the poster design? So basically, I wanted this project to capture kind of my mental state because this semester has been a lot to me. And I also kind of wanted to go into like, I don't know, to me it's like the roots of science, call it what you will, but I wanted it to make it look like I didn't have access to technology. I kind of just sat there, drew everything out myself. It was like scrapbooking almost, but with all of my scientific data. And I also felt like it was kind of more, like, even if you don't like the design, you're kind of like, wow, what is that? So definitely attention. It was a case where all attention is good attention because someone's reading the poster. Biggest challenges were definitely competing with other graduate students for resources, especially when it came to qPCR. Learning the rationale of qPCR and all the other techniques, like what is RNA isolation? How is triazole working? What is cDNA doing? Or how is cDNA synthesis working and stuff like that? Like, yes, people who have done it for a hot second, it makes so much sense because you're like, oh yeah, of course it does that. But for me, who's never done research before, I was like, oh girl, how are these things working? What do you mean reverse transcriptase? What? I don't know. Understanding that stuff and getting past that mental barrier was a journey, but it was fun. I learned a lot. And I guess another challenge was definitely having to do the interdisciplinary aspect of science, connecting to like bioanalysts, biochemists, neuroscientists, to just get answers for different things that I was curious about. What was most rewarding, learning how to code, was I think the most rewarding thing. I've never coded in any of my classes, never coded in my life, and I learned R just by being in this lab and researching. Like every single figure in there was made by RStudio, except for the methods. I made that BioRender, love BioRender, and I'm just very happy with the turnout. I thought it was gorgeous, amazing, perfect. Another thing that was super rewarding too was coming up with future directions and just being incredibly independent on this project. I did all of this basically by myself. Catherine Hamill, who I should be calling Dr. Catherine Hamill to be honest, because she got her PhD. She isn't in this lab anymore, and she hasn't been in the lab I think for like a year or two. So I did most of the stuff by myself. And I'm very proud of myself. I've learned a lot. I've been humbled, but it's been very rewarding. And then as for the future directions, being able to come up with like my own like hypotheses of like, oh my gosh, I wonder if this is connected to that or something like that. Love it. And I hope I can actually do those things. I better do those things, I hope. And then the second question, besides the ones that I mentioned for the future directions, like what future directions are most interesting to me, there are some other genes that I will be analyzing in the upcoming future once I'm done with finals and stuff, where we have genes that are connected to the dopaminergic biosynthetic pathway, and I think looking into one, confirming of qPCR, because, there could be, like, RNA sequencing noise or something like that, kind of similar to ACTA1. So confirming that with qPCR, definitely. But then seeing if we can do maybe, like, a knock-in mice, agonist, antagonist. I'll get there when I get there, but just finding something to do with the dopaminergic pathway. Because I remember from previous studies, or at least there's someone else in our lab, her mice were showing, like, increased novelty-seeking behavior, the 154Q mice. Future research on the project, kind of similar to the above question, but definitely looking into, I want to work with drugs. My true passion is drugs. I and I'm interested in graduate school, maybe like medicinal chemistry or something. If I make it, I still have a year and a half. I'm a junior. But maybe looking into like agonists, antagonists, or like seeing or drugs we have now can kind of like alleviate these symptoms of SCA1. Do it, change every